Hi everyone! This is a short clip on what is an epitope. Let's begin with the types of our immune system. As you might already know, our bodies have two types of immune systems, the innate immune system and the adaptive immune system. And today we'll be discussing about the adaptive immune system, which consists of B cells and T cells. The main function of B cells and T cells is to bind to antigens and to help their removal from our bodies. In order to do that, B cells and T cells have specific receptors. And as you can see here, these receptors differentiate each pathogen by its antigens. And now here comes the epitope. The epitope is the area of an antigen to which antigen receptors or antibodies bind. Antigens are often proteins. Typically, they have many different epitopes that can be recognized by different receptors. In this clip, the receptors are those Y-shaped figures drawn in red, green, and yellow. And by now we all know that those are the epitopes. Speaking of antibodies, which we'll be discussing later, they are able to simultaneously recognize an epitope as well. For that reason, antibodies are efficient when clearing the antigen from our bodies. Now, let's see how B cells and T cells bind to antigens. First, we're going to look at B cells. B cell receptors can directly bind to antigens by their epitopes. This happens in the serum, which is kase in Japanese, or other extracellular spaces. When the antigen and the receptor bind together, the B cell is capable of phagocytosis. And, within a few steps, produces antibodies. These antibodies are also able to bind to antigens directly on their epitopes. Unlike B cells or antibodies, T cell receptors can recognize epitopes that are literally buried within the antigen. But first things first, the antigen has to be chopped into small fragments using proteases. This small piece of epitope then gets delivered to a molecule called an MHC, which is the green block here. Now that the antigen peptide is loaded on the MHC, it is able to bind with the right T cell receptor, which is the orange block here. So here's a quick summary. Antigens are the macromolecules that are recognized by the immune response, whereas epitopes are the areas of antigens where it binds together with receptors or antibodies. That is the end of the video. Thanks a lot for watching.